when I heard about duplex sequencing for the first time. It was like a thunderbolt or a lightning bolt that hit me. We organized a symposium. Tom Wilson cheered us on when he was the program chair at the 2016 EMGS meeting. Stephanie Smith-Rowe and I co-chaired something at the 2016 meeting on using this as a concept. Barbara Parsons spoke. So this is sort of like a reunion. The work that Sharoy talked about led to that formation. We first had the organizing meeting in 2018 for the HESI, a new work group on error-corrected sequencing. A couple of goals came out of that. Sharoy talked about one of the first goals, getting some qualification data for non-clinical safety use in mice and get it published. I'm going to be talking about another goal, and that's to get it into a second species, into rat, and then talk about tech transfer into one or more labs. So we really wanted to start off. The low-hanging fruit is Big Blue. These transgenic rodent mutation assays are sort of the gold standard for in vivo mutation. There's an OECD test guideline. This is what we're doing every day. Our lab helped develop the Big Blue assay in the 1990s through a contract from NIEHS. But what we saw from the mouse data is that duplex sequencing and the plaque data really is equivalent in terms of mutation frequency detection at the C2 gene. That's great if you want to build a new version of the Big Blue assay. That's not what we wanted to do. We wanted really a transformative change in how we do this type of testing, getting more and more types of data to look at. So where it's better is duplex sequencing can really also detect mutations, these synonymous mutations that don't lead to loss of protein function in these phenotypic selection assays. We can also detect mutant signatures, the fingerprint that Jesse talked about. We can also look at the functional class of mutants based on what the changes are. As Sharoy just said, we can detect clonal amplification in oncogenes, something completely different than just hazard identification. But I think the most transformative thing is we can use this technology in wild-type animals and endogenous genes for genomic-wide mutagenicity assessment. It breaks us from the tyranny of using transgenic animals like Big Blue. Even though we set up and helped start validate Big Blue, I'm trying to get rid of it and use something better. So let's talk about rat, going into rat. We ran a bunch of Big Blue studies just to get more historical data. That's where the mouse data that Sharoy talked about came from. So the rat, we ran another study, allometrically scaled doses of the benzoate pyrene, ENU, and vehicle olive oil. We used Fisher 344 rats. These were designed to be OECD test guideline 488 compliant. So we dosed for up to 28 days. We necropsied on day 31. There's new guidance that's just come out. Francesco and I were on a review committee to revise the guideline. Bob Hethwick and others were also on that. More guidance for integrating and harmonizing a germ cell and somatic tissue endpoint. That did not come into this analysis here, but I just wanted to mention that there is a new guideline out there. We looked at liver, a slow dividing tissue, bone marrow, and we included testis. For the mouse, we used a 15 kb gene-based probe set, the C2 gene and 4 endogenous genes with different transcriptional status. For the rat, Jesse really felt strongly that we needed to be more agnostic and not function-based. So we have a new 50 kb probe set with 20 sections over about 24 kb long over 18 of the 20 rat chromosomes. There are 7 that are genic that are shown in green in the diagram below, and there are 13 that are non-genic that are shown as pink. Moving to a new probe set, we were all concerned, what's its performance characteristics? So here's a quick snapshot of the yields we got and some of the quality metrics. I'm not going to go into a lot of detail, but we got hundreds of millions of raw reads per sample, almost 1.5 billion duplex bases sequenced in the rat liver work that we did, with an average coverage depth of over 24,000. Very good hybridization efficiency, so it really worked well. So there wasn't anything that was really limiting going to this larger probe set. High-level types of data that comes out of this is mutant frequency. We're all used to mutant frequency data in genetic toxicology. Here, the individual animal data is shown on the left, the vehicle control, very good reproducibility between individual animals. Benzoate pyrene, ENU, we're getting good significant increase in mutant frequency. There is a little bit of difference between individual animals. We didn't know if that was actually technical variability or biological variability, as Jesse alluded to. So one thing they looked at up at TwinStrand was just taking 2 vehicle, 2 BAP, 2 ENU animals and running the same DNA 3 times. Fairly good reproducibility between the triplicates, but there is some biological variability between the parallel animals between the 2 animals. So that probably is going to be biological variability, just clonality in its liver and its solid tissue. There's probably differentially expressed clones of spontaneous mutations or induced mutations. So that's going to be one thing that we'll have to consider going forward in study design in terms of having adequate statistical power to look at increases over background. So it worked in liver. We moved on to bone marrow and testis. There's a lot of discussion over the last couple of years on germ cell mutations. So we looked at testis. This was following the original 28 days of dosing, wait 3 days, necropsy on day 31. So we had very low expectation of seeing signals in the testes, just because most of the samples at that point are refractory for mutagenesis because they're postmeiotic spermatids. That's another whole hour-long talk we won't go into. But bottom line is, with the new probe set, again, with bone marrow and testis, we had hundreds of millions of raw reads and duplex bases evaluated, good coverage, got good analyzable data. One sample didn't amplify, didn't sequence well. It's probably a DNA quality issue. And we're building that knowledge into how we approach analysis of DNA. We want it to work every time we run it. And so what are the things we look for as we move forward? Another way of looking at the mutation data, again, here's the bone marrow on the left in light blue, the testes on the right, low background mutant frequencies in the vehicle, increased in benzoate pyrene, much higher with ENU and bone marrow. We really expected not to see much of anything in the testes. And these are the germ cells expressed from the seminiferous tubules of the testes. So it's enriching for the germ cell compartment. I was really surprised to see and pleased that we got a good significant increase in mutation with ENU. We did not get it with BAP. We have other animals we're analyzing now with the new design where we dose for 28 days, we wait 28 days expression time, and that will enrich the germ cell fraction of sensitive cells. Hopefully we'll have some new data on that very soon. The data is very rich. This is a way of looking at the performance of each individual probe that was looked at for vehicle, benzoate pyrene, and ENU. We don't anticipate doing this level of granularity when we run commercial studies or R&D studies, but it shows you how you can really drill down and look at this data in many ways. I think the more important thing to do is talk about the tech transfer. How is that working? So TwinStrand analyzed the liver data. They extracted it. We sent them some of our big blue tissues that had been dosed. We'd run plaque assays in my lab. They ran duplex sequencing at their lab. We then brought it up and it's running in our laboratories. And it doesn't get much better than 0.99 correlation coefficient. It really tracked very well at the individual animal level. When you look at it at fold increase over background, another dimension of evaluating mutagenesis, again, very high correlation. One of our samples it was our turn for one of our samples not to amplify well. And it was probably just due to library prep or something. So again, we're working through these early days of having the technology working, and we have every confidence it will work nicely. Now you can look at mutant frequencies. This is just comparing individual animal data, another way of looking at the same data at TwinStrand and MilliporeSigma. Just very good concordance and even the individual animal level variation. So it's probably true biological variation and not technical issues in terms of reproducibility. You can look at things like simple spectra. This is just looking at the individual transition transversions using unsupervised hierarchical cluster analysis to group them by similar patterns. We were able to pull out the ENU vehicle and BAP from our our data that matched exactly what TwinStrand did. So it gave us a nice confidence in what was going on. Grouping it to 6 different transition transversions that could be going on in the DNA. This is just at a macro level showing what TwinStrand got with the benzoate pyrene light blue, ENU dark blue, light gray vehicle replicates in our data too. So it's just giving us the confidence that we can do the assay really to the same level and rigor as the masters at TwinStrand have it up and running. We talked about trinucleotide spectrum. So this is the, instead of the one overall mutant frequency, this is breaking it down into, the 6, C to T, the individual transition transversion, but then adding on the flanking bases. So this is the trinucleotide context, the local context for what is mutated. And there's very unique specificity by what's going on. So here is the vehicle control. This is probably just the junk that happens with aging, deamination, things like that. But we were able to replicate, this is the group average stuff at TwinStrand. This is our individual animals, just showing there's good reproducibility between individual animals, not just burying variation in a group mean. So we're very happy with that. This is going to the ethyl nitrosourea spectrum. So if you go back, you see there's almost nothing over here in the T to A, T to Cs, T to Gs. Very unique spectra here with the nitrosourea, very potent direct acting alkylating agent. We're getting these peaks here that you don't see. Again, we replicate that pattern among our various animals. Now look at what the pattern is here for the nitrosourea. Most of the mutations are here. Going to a polycyclic aromatic hydrocarbon that requires hepatic metabolism. Here's your benzoate pyrene. Nothing over here. So this is the basis of Ludmila Alexandrov's patterns in the cosmic database. This is the power, I think, of what this assay technology can do. It will show you a different dimension of just straight up overall macro level mutant frequency. You can really go in and look at the nuances of what is going on at the base level. Clint Valentine up at TwinStrand has been excellent to work with. He's really a numbers jockey. He did this cosine similarity. It measures the similarity between 2 n-dimensional vectors that's sort of independent of the magnitude of the signal. It's one way of comparing our fairly complex trinucleotide spectrum, but as you get your arms around it on comparing one trinucleotide spectrum to another. So we could probably be using it for our vehicle versus treated. But here we are comparing, say, our ENU, MilliporeSigma ENU against TwinStrand, very high correlation, benzoate pyrene. The vehicle is not quite as high. The counts, the coverage depth in these initial runs may perhaps be higher, but we still were able to pull out those correlations fairly well. But again, these are the lessons learned that if we want to start using trinucleotide sequence, we can run the numbers backwards in terms of coverage depth, DNA loading, to give us the numbers for adequate counts per bin. At the individual animal level, I'm not going to go into a lot of detail here, but you can see again, the ENU, very potent mutagen, strong correlation. Benzoate pyrene, also strong. A little bit of an overlap of patterns between BAP and the vehicle. So it wasn't quite as tightly pulled out. And again, the lower coverages here on the vehicles is why this is not as strong as it could be. But anyway, this will give you a sense of how I think we're going to be approaching data analysis in the future. Now, this is where it gets interesting. We're not envisioning that this will be part of a standard baseline error-corrected sequencing mutation analysis study. But if you're testing an unknown and you get a positive, we can go and ping against the Sanger Institute in the UK, their cosmic database, and look for affinities. So here is the vehicle control. It aligns best with Alexandrov's cosmic signature 1, which really is representing normal deamination processes related to aging. So this is the bad things that happen as you get older like me, but it shows up. Benzoate pyrene with the yellow box up here, it aligns as Shiroi mentioned with signature 4. which is associated with polycyclic aromatic hydrocarbon exposure, human lung tumors. I should mention the COSMIC database is an aggregation of tens or hundreds of thousands of human lung tumors, and what signatures are seen, and if there's an etiology associated with it, you can map it. When you get to E and U, humans don't get exposed over a lifetime to nitrosoureas, so there really probably is not a strong signal within the COSMIC database, but it does align with 4 different patterns. So again, a signature for this type of mutagen is clearly there. So let me wrap this up. So we approached this as the plaque-based Big Blue mouse and rat assays are sort of the gold standard. This is, I'm running dozens and dozens of these a year in my lab here at BioReliance. I think there's a better way to do those things. So we showed in the mouse data, and now with this rat data, or actually we only did the C2 gene in the mouse, but duplex sequencing is equivalent to this gold standard plaque-based assay in terms of mutant frequency, mutant spectra, functional class, all the attributes that we can drill down into using duplex on top of the Big Blue. We demonstrate that we can work in multiple species, so we have both mouse and rat working. We moved into the TG RAS-H2, so we have a non-transgenic rodent mutation model. So being constrained to using Big Blue, Mutamouse, GPD Delta, we can actually walk away from that at some point when we convince enough people that this is working. It's robust. As we stepped out from liver and bone marrow into lung, spleen, and blood with the RAS-H2, as we went into testes with the rat, it just held up and it kept working. We switched genomic panels, so it seems to be probe independent. From a hazard ID perspective, DNA is DNA is DNA. You pick your probe, you make sure you don't have any weirdness in your probe set, like highly repetitive areas and things like that. So from a hazard ID perspective, it's pretty straightforward. If you're looking at oncogene activation, you don't want to be cutting out the multiplicity of unique independent events that may not be independent of clonal expansion, but that's for another day. And it's working with multiple mutagens. I think the key thing is it's not a hothouse flower. It can be moved out into other labs. So we were able to move it out into our lab, Health Canada. I know other labs are doing it, FDA, NIEHS. So you'll hear more from other labs also. I'm speaking from our experience here, but we could really solidly replicate the DNA, the TwinStrand data. Good correlation in terms of mutant frequencies and spectra, which I think will be the foundation for hazard ID, whether integrated in as a biomarker of cancer risk in, say, 6-month rat toxicity studies, early repeat dose studies and drug development, to resolve positive in vitro gene tox assays, as we do with gene tox right now, or even as an option 2 under ICH-S2R1. A lot of options are there. And of course, we can map it to the COSMIC database. So, where are we as a company? We've kicked the tires. We believe in it. We've worked closely with TwinStrand. We're planning a commercial launch. We're actually talking with companies right now for early R&D validation studies. So let me just say thank you to TwinStrand, to Jesse, to Clint and the team up there, to Mark Fielden, who had the vision to bring us together in this collaboration and to help support it, to Shroy taking over for Mark, but then people like Barbara Parsons, who had a similar vision, for Christine. So I think we all kind of see the same opportunities. So with that, I'll stop. And I think we're probably short on time, so we'll probably take questions at the end. Thank you very much.
Thank you, Bob.